Thank you very much, Mark. I'm more here to do a bit of introduction, and then I'm going to pass over to my master student. He's done some really great work in the last year. So,
talk to the microphone, if you would.
Can you hear me without the microphone, or do you? Oh, okay.
Let's do that. You can go. There's one on the left. The one on the left of the screen. That's it.
Better?
That's fine. That's good.
Cool. So I'm industrially funded. So I'm a PhD student in my final year, funded by Coca-Cola Hellenic. So starting to move the research up into industry. So I'm industrially led, and I'll let you introduce yourself in a second. But I just want to say that it's been really nice to be here today to listen to it's part of the story that I don't get to do. So I'm more focused on when you've recovered it, and you've been there and you've treated and you've recovered all the plastic, or whether it's PE, PET, what happens next? So all my work has been on PET and that hydrolysis using those enzymes and biorecycling and the future of that. Is it the solution? Is it going to be the standalone option, or is it going to be working collaboratively with other options as well? It's really interesting. Amy's not here, but she made a comment about obviously the greenhouse emissions. All our work that we do now in the CI and the research labs is all led and directed by the techno-economic analysis and a life cycle assessment. So everything we do is now targeting research areas which are flawed or needing optimization as a result of the analysis with regard of the carbon emissions. So it's very important. But yeah, so I'm going to let you take over.
Yep.
And then questions later.
Hello, everyone. My name's Haydon. I'm a master's student at the University of Portsmouth. And I've been spending the last few months looking at plastic degrading enzymes, and more specifically polyethylene. So I'd like to talk to you today about why we believe that enzymes may be the solution for increasing plastic pollution, including nodal spillages. So I was going to start a bit on the background. I don't think we really need to do that after everything today. So I'll go a bit onto the Express Pearl. Which again has been mentioned, but this sank off the coast of Sri Lanka. It was carrying 25 tons of nitric acid. This was not safely secured, and the Express Pearl attempted to unload this in ports in Qatar and India. Due to logistics they were unable to do so and continued on their route, where the tanker eventually sunk, releasing 1,680 tons of these plastic pellets, and also toxic chemicals that were extremely damaging to the local ecosystems. So this brings me to the issues of waste management. Being that these were collected off of beaches, there's a large amount of physical contamination. And then there's also the side of chemical contamination. This comes from the combustion products from the fire on board the ship, as well as the number of toxic chemicals that were leaked. These have a tendency to leach into the plastic, and hence these are assumed to be hazardous. Because of this, they are highly restricted from being moved out of the country by the Basel Convention, of which Sri Lanka has signed. And hence, the only options are internal, of which are highly limited. And this leads to the majority of this waste being kept in these secure storage containers. So if you look at this image, you see a lot of organic matter, trees, plants, there'll be animals living here. If you think all of these are made up of organic polymers, when they die, they don't stay there forever, they're decomposed. You might think it's the animals and insects and microorganisms breaking these down, but in reality, it's the enzymes that these produce that are able to break down these organic polymers and create a nutrient cycle. So in conclusion, nature wastes nothing really. So an example of this is the cutin in wax leaves. These contain ester bonds, and they can be broken down by enzymes called cutinases. Protein, this contains these amide bonds, and they can be broken down by enzymes called proteases. And when we're looking at the plastics that we usually work with in the labs, with PET and nylon, you can see these same bonds. And similarly, these enzymes show activity against these plastic types. and are able to break them down. That brings me on to what we do at the CEI. We have a discovery team that is currently, I believe, in mangrove swamps in Asia, and they take genomic samples of soil, water, and directly off of plastic, attempting to identify any sequences that may produce promising enzymes to degrade plastic polymers. From here in the lab, those that seem promising, we can produce, attempt to characterize so that we can compare them to other enzymes, and the promising ones enhance. We can also look to enhance enzyme production, say by changing the organism host producing these enzymes, and sort of optimizing the conditions of which they're being grown in. We also have deployment, which is where we mill our own plastics to be similar to industry conditions, and we look to degrade them in 250 ml or 1 liter bioreactors, which are sort of a scaled down version of what would be used in industry, where you can have heating control, stirring control, and pH control. And then we also have apply, which is mainly what Brooke does. She looks to break down large quantities of these plastics in those bioreactors, purify off the monomers, which she works with PET, so they're terephthalic acid and ethylene glycol, and use these to repolymerize plastic of a virgin-like quality. And then also industry, where we talk to industry to see if there's any conditions they would want these enzymes sort of optimized for. So that brings me on to, we initially believed these nurdles from the Express Pel would be PET. We later completed DSC, shown here, to identify the melting temperatures. And we found these to be 2 types of high density polyethylene by that melting temperature, and not PET, which we would expect to show up quite a lot higher. So this is not surprising if we consider the density of seawater. PET is a lot more dense, and polyethylene is less dense. So in these cases where large quantities of nurdles are spilling up on the shore from off the coastline, it is assumed that in most cases these will be buoyant polymers such as polyethylene and polypropylene, which is a bit annoying considering PET has this ester bond with this Achilles heel that our enzymes at the moment can break quite easily, and we can show near 100% depolymerization of around 16.5% weight to water of PET in a reaction. So the challenges of polyethylene and its carbon backbone is that the carbon backbone there being carbon to carbon, it shares its electrons equally throughout, and this means that it is essentially non-polar. This means that the polar molecules that usually forward reactions have a difficulty sort of attacking these bonds, and it is essentially greasy, which is compounded by the carbon and hydrogen bonds sharing their electrons relatively equally. So you end up with long molecules like this with no polar charges and is essentially greasy. And then these acids, bases, amino acids, and water molecules that the PET degrading enzymes normally use to break apart bonds cannot easily interact. Brings me on to what can degrade these plastics. It's been identified in papers before that these waxworms here are able to chew through polystyrene, polyethylene, and polypropylene, all hydrocarbon plastics. But interestingly, this wasn't just a mechanical action. This was the saliva and the enzymes within it, especially in the case of polyethylene, being able to degrade this plastic. It was thought that this activity has come from, is carried on over from the waxworm's natural food source of beeswax, which contains these long carbon chains similar to polyethylene, although we're say just one ester bond. From here, papers has also been completed allowing us to identify the structures of these enzymes, which I'll just show here now so you can look at them. There's Cora, Ceres, and Sibelius Demetra, which is made up of 2 separate proteins. So when we're looking to produce these enzymes, they're originally produced in these insects. However, due to the cost of this production system, the amount of protein that you can produce is not very high. It's difficult to engineer or improve these enzymes when using these systems. We will not be using these. In our labs, we commonly use bacteria or E. coli, although in the case of these enzymes, they are eukaryotic, produced by insects, and they contain eukaryotic modifications on these proteins. Enzymes being bacteria being prokaryotic cannot produce these modifications, and hence we won't be using these in case that affects the activity and structure of the enzymes. They just might not work, essentially. So that led us to the use of yeast, which is eukaryotic, and specifically Pichia pastoris, which has previously been used to express a number of proteins. For example, the hepatitis B surface antigen used in vaccines. How do we actually get from waxworms to yeast? Originally, we identify the DNA that is encoding for these enzymes. We can optimize them for yeast, so they essentially have their own separate languages in which they produce these proteins. So you can run it through software and have it optimized for yeast, and then we place them into these expression plasmids. These contain an antibiotic, these produce an antibiotic resistance, allow us to secrete the proteins out of the cells. There was one other thing. And allow us to trigger protein production on the addition of methanol. From here, we can then amplify this DNA using bacteria, purify this, and insert it into yeast, which we do so by basically electrocuting these. From here, we can plate those transformants or yeast-containing the plasmids and spread them onto agar plates containing an antibiotic. These yeast, having hopefully a resistance for this from the plasmids, should be able to survive, and those large colonies are those which we expect to have best uptaken the plasmids and should hopefully be producing these enzymes. From here, we'd run a number of small-scale growths and protein expression tests to try identify those colonies that are producing the enzymes, and we look to use them to upscale in large flasks and these 1-liter bioreactors. From here, we can separate off the cells and purify the liquid that they were grown in to obtain these pure enzymes, which we can then use for further experiments. For the results and outlets, you see this is a sample of purified DNA which I made. You can barely see it, but that tiny little water drop looking thing there is 100% pure DNA. We can then use this to spread all of these plates, which we've done before. And that's given us, in this picture here, 2 colonies shown to be producing this protein, Sibelius, which is one of the 4 proteins in the waxworm enzymes that make up the 4 waxworm enzymes. We have also seen production of Cora, one of the other proteins, but to a lesser extent and not as clear as in this picture. So the outlets, this is the first proof of concept that these enzymes can be produced by yeast rather than the waxworms, which we are not sure that was even going to be possible. It's also important for future research efforts, being able to produce these at larger scales we'll be able to complete a number of different tests that haven't been conducted. And also, being that these can be produced by yeast will increase the ease of engineering these enzymes in the future. And again, for industry, this is important, being able to produce the large scale of enzymes that would be needed for any application. Again, this being quite early stages, it will increase the knowledge in the field of PEases, and could be a potential first step in a solution for enzymatic spillages of polyethylene and also polypropylene plastic nodal spillages. And this could be potentially useful for the whole plastic waste management industry, being that actually nearly 3/4 of the plastics being produced currently are carbon to carbon backbone plastics, which don't really have a lot of coverage in the literature right now. So the next steps would be mainly to characterize these enzymes, so allow us to compare them and their conditions of which they prefer. So for us, that could look like identifying the temperature of their unfolding and the time it takes for them to unfold at certain temperatures. So sort of analyzing their stability along a broad temperature range. We'd also look to assess their activity against polyethylene and compare that to the insects, that the enzymes produced by the insects, and also identify the optimal conditions such as pH that these enzymes work at, which still has not been completed. And also thinking about storage conditions. So in this picture here, this is protein that has precipitated out of solution. And once this happens, it's very difficult to recover that. And this is something that we've seen trying to upscale the production. And again, it will be something we'll have to look at in the future and consider. We'd also like to design an activity assay method and ultimately engineer these enzymes to increase their activity against polyethylene. I'd like to give a big thank you to my support team at the CEI, my supervisor, Andy, my co-supervisor, Bruce, the tech transfer officer, Louise, who sadly can't be here because she's injured, and Brooke, who's been assisting me with day-to-day lab work. At OSRL, I'd like to thank Ria, who isn't here, also Marta, Rob, and of course, Mark. Thank you. Do you want to come up?
So you have an industrial project, you have an industrial trial coming up.
Yeah, so
Sounds a little bit about.
usually there's lots of questions you get about whether or not you're going to release enzymes into the sea, is it going to break down everything. So I'm surprised I haven't got as many questions as we thought. But yeah, so
If you can answer the one on the industrial trial.
so the industrial trial, so polyethylene, this here is the first sort of scope and work now being done with polyethylene. There's another team up in Nottingham, which is now working alongside us. But in regards to PET, as Haydon mentioned, with those ester bonds, it makes it easier for us to break down, but it's a natural bond which is found for us to break down. So that research has been going on for a couple of years, and there's a company in France called Carbos who have taken the technology, which was first discovered back in 2016, first enzyme, optimized in 2019, so it's relatively new. So they took the technology and the theory and they made it commercial. They tried to stay quite independent from the research. They took the idea and said, right, we're going to make it industry, we're going to make money from it. What happened with that, like any process, mechanical recycling wasn't perfect the first time round. It's taken years and years to get this efficient, streamlined process. The same thing is now happening with this biorecycling. So in theory, it was proposed. In practice in the lab, it works. It's now being scaled up. So in industry now in France, next year, 2025, they're opening their first large-scale pilot plant. So it's already been built now. They've already released a couple of papers in Nature, which is what all our research is based off of. And then following the establishment of the process, they then reached out back over to us because they realized it wasn't as easy as what they thought. So now there's certain hurdles and challenges, like with any process, which we're now trying to mitigate and overcome. Things like the crystalline structure of PT, that poses challenges for us. There's certain ways you recover the monomers. So to recover TPA, which is one of the monomers, the building blocks of PET breakdown, we crash it out using acid, which obviously at the moment isn't ideal, but other ways which we can do it, which is more efficient and environmentally friendly. So at the moment, the technology isn't just an idea, it is starting to be scaled up. The benefit to this is obviously with linear recycling. So I know the comment earlier about recycling technologies. I agree, the current way we're going, we cannot keep going in the linear method. Linear recycling is not sustainable. However, this is the first scope of a circular plastic economy where the emissions are low, so it's green. So enzymes are natural, there's no byproducts which aren't what we want, which is the virgin materials. And those virgin materials, because we've chemically broken them down, are building blocks then make the same strength plastic product. And that's where the patch, that's where this is kind of gold star, is because we can actually take a plastic bottle, hydrolyze it back down, extract the monomers to a purity which is actually of equal or more pure than what we're buying at the moment, and then repolymerizing them back together to make the same strength, because it's chemistry, it's taking it back down. Well, there are emissions in the process, like anything else, but it's weighing up like the carbon one. That answers the industry. But yeah, sorry, question from the crowd. Yes.
Yeah, I have a question. Sorry. One of the things we've observed on the River Itch in particular, which is a chronic issue there, is that pellets get incorporated into the kind of upper 5 to 10 centimeters of soil material, and you've got weeds sort of amongst it as well. Do you see this as potentially being applied in that environment? Because from a cleanup point of view, we're not going to go in with excavators and remove that. So could you apply this in situ to address that sort of contamination?
Enzymes themselves, I wouldn't apply it directly to the soil. That wouldn't work. Enzymes quite fussy. So I assume everyone here knows an enzyme, and we shouldn't assume that. So enzymes, small proteins, they're quite fussy. So they need to be in a buffer. Sometimes it can be water, but they're pH controlled, they're dependent on temperature. So to maximize the efficiency activity, we need to do it in a controlled environment. There are avenues which people are working on of involving the actual bacteria themselves and microorganisms. So one approach would be to release those microorganisms back into the soil. However, that poses a threat and the ethics of your introducing something which wasn't meant to be there. The slide Haydon showed of those small gel proteins, I try and highlight every time that we act like God. You're literally taking a yeast cell and changing its DNA to produce something that it never ever was meant to produce before, which is great in a lab. But I personally wouldn't release that out into the world because you don't know, as you mentioned earlier, what the consequence of that would be. So I'd say there'd be a lot of studies to see the scope would be there. You could take a soil sample in the lab and try it, release bacteria in there, and that bacteria would work its way through. That's where it came from. I mean, 2016, I don't know if you guys have seen it on the news, but that first enzyme came from bacteria, which was found in Japan on a waste recycling dump. So it was already there, starting to break it down in the soil. They thought it was bacteria itself munching away, but it was the enzymes, which just took the catalytic mechanism, applying to the cutin, applied it to the bonds. So it needed a study to confirm it had no negative effects. Okay. Professor.
Have you ever contemplated doing a remediation in situ or on sort of floating islands where you get bioaccumulation or accumulation anyhow? It creates naturally an ecological niche. And well, that's the question. Have you, they're sort of,
Do you mean
almost like terraforming? Like you put the creatures in that you suspect would just like that and continue forming it. Because there's so many sort of sensitive lands. If you literally trample through it,
you're introducing,
like there's sort of estuarine areas where one could remediate the water body.
Really good idea, actually. I hadn't personally thought of that. We take it up to our director. Do you mean taking kind of like a film, putting it on a floating island, or creating its own? Do you mean creating an own floating island of the bacteria, treating the plastic with it?
We have seen it happen naturally, that you get sort of quasi-terraforming things, floating islands. And sometimes they actually have floating reed, but reed is a problematic thing. It creates so much biomass that it goes back into the environment. But there's sort of observable concentrations, hotspots. On land, I've not seen it, except maybe sewage remediation on the old meadows, as it used to be done, and then it sort of creates an ecosystem there. But you could, with your knowledge, basically accelerate the adaptation to what is fundamentally a nutrient stream.
Yeah, I know we are doing kind of fast track evolutions. We are taking kind of bacteria and forcing it to grow on substrates that it shouldn't grow on, and taking the best ones and then making them grow again. But you know, I like that idea.
So
I'll look into it.
we're now missing Chernobyl, but that's a whole different.
Quite different.
Kettle of fish. But you get nature's incredibly inventive, not very fast,
But it is.
yeah.
But yeah. If you have any questions?
Any other questions?
With the enzyme work, how pure does the feedstock of plastics have to be? So can you put in mixed plastics, or if you're recovering plastics from the environment, do you have to do a massive sort to make that polypropylene as pure as possible that goes in? Can you have bits of polyethylene and bits of PVC and that? What will that cause?
So that's one kind of avenue of research. You can imagine there's loads of different kind of pipelines going through. Enzymes are very selective, so it's substrate specific. So that's the beauty of it. It would need to be sorted initially. However, kind of industrially, with my kind of work that I'm doing, speaking with the kind of plastic producers, they've actually got a lot of hub sites already established of kind of the sorting, washing, the hot flake production, the pelletization, and they're actually more interested after their assessment for us to target the hot wash flakes rather than targeting kind of mixed waste. The dream in the future would be to be able to take a massive mixed waste stream, put in 10 enzymes, each targeting different things, extracting different stages. At the moment, it'd be a case of we keep the sorting and collection just because it's streamlined at the moment, and because of money and things, it would start off that way. But in the future, most definitely, the kind of goal and the aim would be the fact that you can have a process that enzymes specifically would target different plastics at different stages, and you'd extract the monomers of each one at different points. But that is years in the future, but it is a feature which could be there, which is the hope.
Lovely.
Sorry, just to kind of come back to some of the other presentations earlier today. What's the economical impact of creating the enzymes, and you know, does the outcome outweigh that
Benefit. So in my opinion, yes, and backed up with kind of the data aspect. So you've got one aspect looking at as the situation we have now and the future and the options we have. And if this was optimized, it would give us a completely circular plastic economy, which is what we're all trying to aim for, which is green and sustainable. The streamlined process of enzyme production is already there. We already know how to make plastic. We shouldn't be demonizing plastic, as someone mentioned earlier. It's not going away. It's how we deal with the waste mismanagement. That's the issue. So that's factually need to improve that. So the impact of the research. So very quickly, back in the day, you'd have whole kind of people's PhDs solving the structure of one crystal, one protein. We now have so much AI and so much techniques that you can do that in a couple of weeks, couple of months now. Everything's advancing. The enzyme design that we do here at CI for plastics also informs kind of research on other things. So we kind of work with DSTL looking at some of the really nasty chemicals, because everything is biology, which is chemistry, which is physics. Everything in theory can be broken down. So let alone the aspect of plastic recycling and the benefits of working in the industry, looking at research, the benefit is that everything we do informs the field, which informs other waste streams and problems that we have, not just plastic. There's a lot of problems as well. Do you want to add any comments?
Good.
Or not?
You don't correct.
Yeah.
I mean, moving on to the questions.
Thanks very much, Brooke. Thank you very much, Haydon.
Thank you very much.